Also now, just recently started this really amazing project called FathomNet, and sort of as a larger umbrella, Ocean Vision AI. And it's really trying to build up this sort of system and the community and the data so that we as computer vision researchers can actually contribute and sort of help try to understand life under the ocean. And I'm really excited to see what she's gonna be talking about today.
That's very nice of you all. Well, so I'm very excited to be here. This is only my second CVPR ever. And last year I really just exclusively talked about FathomNet. And this time around, I'm going to try and give you kind of a survey or a sampling of some of the activities our lab is involved in, but also some of the questions that we're really interested in. And I also want to point out, right, we aren't computer vision researchers. We're really practitioners. We like to use what you all as a community create for our use cases. And so while I show you some of these activities, I really want you to think about where might be some ways that the computer vision community could potentially interact with us, because we're sitting on like massive data repositories, for instance. But first, I need to highlight the group. I'm actually very fortunate. There's a lot of us here. I see Yost. But Yost, Lily, Eric, and Giovanna are big members of, Kevin Barnard are big members of the Ocean Vision AI effort, but also FathomNet. So you'll run into them. We're also doing a FathomNet workshop on Wednesday. I'll have a little more information on that. But because of them, we've got all these active projects. A little bit about MBARI, because I always get questions about what is that institution. It's a really, really small research institution. It's based in Monterey Bay. Obviously, it's in our name, which is a really short 1.5-hour drive between like Stanford University and our institution. Where we're located, it's very strategic, actually. You'll see if you look at the bathymetry maps here, we're really, really close to deep water, where within about an hour sail, we'll get to water depths of about 1,000 meters. So from a scientific perspective, as well as an engineering development perspective, that's really important because you can quickly test things, turn things around, make adjustments, and repeat the process. Please come in, and I know there's not a lot of space. Feel free to sit. And what MBARI does, what we're really focused on is the development of tools and techniques that allow us to understand the deep ocean better. We were founded by David Packard, which I think you all have heard that name before. And so that is really our mission and what we're focused on. And at the institution, we use a lot of different robotic systems. So on the left, we have remotely operated vehicles. This is actually Falcor, but we have 3 different remotely operated vehicles. One of them is rated to 4,000 meters. And we also have a lot of autonomous systems. So these are robots, these are drop cameras, but a lot of these systems are rated to 4,000 meters. So access is the game, and we essentially have that, fortunately, at MBARI. But what I also want to point out is this actually describes kind of the evolution of the activities and projects that we've had in the lab. And I will explain that in a little bit, but really I want to focus first on how we've leveraged these remote vehicles to do some really fine scale quantitative imaging of animals in the deep sea, and then how we're now using autonomous systems to really scale our capacity for observations of ocean life. I have to show you all a video of deep sea animals, and I've highlighted my favorites. If you wanted to check out some more, you can go visit MBARI's YouTube channel. It's kind of insane. The top left, and I always have to mention this in any talk I give, is a giant larvation. And I don't know if any of you have heard of this kind of animal, but the animal is that kind of a, looks like a larvae or tadpole. It's what's beating its tail, and then it secretes mucus around it, and it lives inside that. It's a really complex filtration structure. We call them snot palaces for fun. We also have calicophora and siphonophores, and this one is Praia dubia. I don't know if we can turn the lights off, but basically those animals, if they were considered an individual, they would be the longest animals in the ocean, not the blue whale. What you see here in the bottom right, those are midwater worms. We think a lot about earthworms and what they do, but the animals in the midwaters, the worms especially, are completely different. In this case, this worm is a Tomopteris. It swims really rapidly. It can actually stop instantly by arresting all of its legs and then reverse itself by just changing the direction of a body wave that it moves along its legs. So from a robotics perspective, they're really, really interesting for bio-inspired design. And then finally, last but not least, the benthic communities that include, corals, sponges, etc. So it's all of these groups that we have ready access to, especially in the Monterey Bay area. And then high level, I just want to explain why we do the weird things that we do in the lab. And the idea is that if we could combine these three things on the left, right, the creation or use of innovative observation platforms, create state-of-the-art instrumentation that we can affix on those sorts of platforms, and then we will then achieve not only access to novel study systems, but then through collaboration, this could be with biologists, mathematicians, physicists, really delve into deeply how these systems function, how they work, and how that might actually inspire other sorts of technologies. So this theme is bio-inspired design. So that is what the group is largely focused on. But here we're spending a lot of time thinking about how do we do all of these things really well, and then engage domain experts to use these systems and answer their particular questions. So to start, I want to show you some imaging systems that we've developed for our remotely operated vehicles. I'm going to highlight a few systems that are already on board these vehicles so you could think about different ways to interact with this kinds of information. So first, there's a science camera. There's, in some cases, multiple cameras that are integrated on these vehicles. There's also collection containers, so they have ability to not only collect animals, but also bring them back to the surface to do all sorts of lab measurements. And then we also have an auxiliary manipulator. So what this allows us to do is, do collections, maybe experiments with systems that we're trying to study while we are deployed in situ. I've also, in this photo, we have a VR camera system. We've actually collected lots of data for this system. So if you're interested in looking at that, I'm happy to provide it. And then three other imaging systems, which I will briefly talk about for this talk, is a stereo camera system that we've used for our machine learning integrated tracking algorithms for these vehicles. We've also developed a laser illumination system called Deep PIV. So it's a laser line scanning, essentially, that allows us to do some observations. And then last but not least, an instrument called IRIS. It's a light field imaging system that gives us like three-dimensional depth data at 60 frames per second. So with all of that in mind, I'm going to quickly go over DPIV and IRIS. And then with my remaining time, I'm going to try and spend a little bit of, time going over some of the autonomous systems and what we're thinking about when we move into the future of ocean exploration. So the way DPIV works, it's actually attached to the manipulator arm on the vehicle in this case. And there is a housing that you can't see that contains, I think, a 2-watt laser that is injected into a fiber. And then there's some optics in that housing there that then creates a sheet of light. We use red light because that actually helps to reduce animal response to it. And so we illuminate a sheet of light in front of the imaging system, which is, I think, HD resolution at 60 frames per second. And the example, I should also add, the field of view can vary because there's a zoom lens. The depth rating is 4,000 meters. And really what I think sets this system apart from other systems that may have been deployed in the ocean before is the fact that our mission duration really is only limited to how long we put the vehicle in the water. Some places that could mean 24-hour operations for collecting this kind of data. Also, what we do is there's a dye and particle injector, and what that allows us to do is inject some sort of scatterers into the water just to get a sense of what sort of interactions we might expect, and then go in with this laser system to do actual quantitative measurements as well. And some highlights, we've been able to use DPIV in a scanning mode, so a lot like MRI or CT scans. Remember, we're just illuminating a sheet of light. So as you move that sheet of light through an animal, a gelatinous animal in this case, or even the mucus house that it resides in, you're getting individual planes of information that you can reconstruct using image segmentation, a lot of approaches that this whole community is very familiar with, and get three-dimensional reconstructions of these systems, not only getting exterior views of what these structures might look like, but then, and I feel like I can say this, unprecedented views of interior structures inside these snot palaces. And what you'll notice is the complexity, right? There are all of these chambers that go off that central chamber. And what's interesting is they all connect in one location near what we call a buccal tube. That is essentially a straw that the animal puts in its mouth. So these are elaborate structures that the animal is able to create by just secreting mucus and beating its tail to blow them up. And from an engineering perspective, like how on earth is an animal like this able to do that? And then do so rather repeatedly. I think we have guesses that they make these mucus structures on the order of maybe once per day. We don't know that because we haven't had those kinds of observations yet. Another example of how we've used the imaging system is to actually measure the movement of suspended ambient particulate and use that motion of the particulate as a proxy for motion of the water around the animal. This process is called particle image velocimetry. It's a very common technique used in mechanical engineering or fluid mechanics labs around the world. But the idea is that now we're able to do these kinds of measurements, and I apologize because this looks much better in a very dark room. But when we take the instrument and we position the instrument on the animal is what's beating its tail there. And then that outline you see around it is the cross section, right, of that mucus structure. And if you look closely or squint, you can see particles. Wow, Kevin, thank you. I might play that again. I think I have time. So not only are we getting the cross-section of the animal within its mucus house, we're also getting the displacement of particles that are suspended around the animal over time. And so using this particle image velocimetry or particle tracking velocimetry approach, you can directly measure filtration rates or volumetric flow rates of what this animal is processing when it's beating its tail. And so we've been able to do these measurements. Now remember, these are all in situ. This particular animal is 400 meters deep. It's about 2 centimeters across. The mucus structures can be up to 1 meter across. And we're having I think it's a 6.5 ton vehicle position a 1 millimeter thick laser sheet on an animal that's about 2 centimeters wide. So the fact that we're even getting this data is astonishing. But then also the level of detail that we're getting from just this one imaging system alone is, I was very excited the first time I saw this. So this is at least the DPIV instrument. It's focused on, it's a planar measurement, right? So it's really good at giving us 2D measurements or 2D particle motions. But let's face it, the ocean is a massive 3-dimensional space with a lot of complexity. Animals cannot be, simplified into 2D structures, although physicists would love that. And so what we need are systems that really give us volumetric data or 3-dimensional data that we can then use further. And so we've done a very, very intensive and comprehensive look at all of the mechanisms or approaches that you can take for collecting that kind of 3-dimensional data. And then given those approaches and then our limitations for trying to get an imaging system like this on a deep diving vehicle that's rated to 4,000 meters, we landed on planoptic imaging or light field imaging, which I don't think I need to explain too much to this audience. But right, the approach is, at least for a single camera light field, you have a micro lens array between the image sensor and the imaging lens. And what that does is effectively subdivide your image into multiple perspectives that you can then use to come up with volumetric data. So the way this will work is this is a raw image collected from the camera system. Each one of these dots corresponds to a single perspective within that micro lens array. And then using this information, you can come up with depth maps, right? Colors indicate distance between the object and the image sensor. And then you can, I think what I love too about this process is given that depth information, you can also create what we call total focus images where everything is in focus. And then finally, you can get three-dimensional reconstructions of the entire, in this case, particle field. And so this is a imaging system that we built, or we started building, I think in 2018. It's called IRIS for remote imaging system. And the way this works, we have, again, using the ROV manipulator, you have the IRIS imaging system. It's a single camera housing or single housing, which is very different from DPIV, which is 3 housings to deploy. And we have, we're using a Raytrix light field camera. That's a company based out of Germany. We have currently a zoom lens, so our imaged volume can vary from 5 by 5 by 2 centimeters cubed up to 12 by 12 by 6. So still relatively on the small scale for some of the animals we like to study, but a really, really good starting point for us. Depth rating is 4,000 meters. And again, we're using red light to try and minimize animal behavior changes. And then another thing I want to highlight is we have this 25 megapixel camera that takes data at 80 frames per second. So we have data rates on the order of 2 gigabytes per second. So it's lots of data that we have to manage. And then very recently, I think this was last August, we were able to deploy this system at a seamount called Davidson Seamount, which is off the coast of California, but a specific site called Octopus Gardens. And there is a location where you can find hundreds of the same species of octopuses that are brooding. But besides brooding, they're also crawling, like you see this particular octopus is doing, and then also free swimming. And so when we went over this 12-hour dive, we were able to collect IRIS data of all sorts of different octopuses, individuals doing all sorts of different behaviors. And so we're kind of sitting on a big data set we haven't fully processed. But here is a video showing you all how we position our instrument relative to our particular subject, in this case, a crawling octopus. And try to position ourselves relative to the animal, so we're getting the kinds of data we need, but also trying to minimize those interactions, right? We really don't want to change the behavior that the animal is eliciting while we're trying to measure it. And so what this is the depth map from the imaging system, IRIS, and we're collecting data. In this case, I think it was at 60 frames per second. So you can see the level of detail we're getting from this image system. And then what we have to do is, markerless tracking. We can't put anything on these animals to make it much more easy to do feature extraction or tracking. And so, unfortunately, Yoast spent a lot of time looking at this footage, selecting different kinds of features like the suckers on the arms, and actually for the first time being able to do, three-dimensional reconstructions of tentacle movement during crawling in situ of octopuses. And if you look at the literature for like octopus-inspired robots, most of those robotics papers are, and we're talking hundreds of papers, are derived from 2 to 3 in situ studies of octopus behavior. So already the kinds of data we're able to collect here really will, I hope, kind of change and improve our capacity for generating this kind of information of animals in their natural environment. The other thing that we haven't had any time yet to pursue, but is next on the docket, is looking at the outputs for particle and animal fluid interactions. So the idea here is we're getting three-dimensional particle representations within the scene, and that combined with the surfaces of the animal itself can give us an indicator of like how does the animal movement affect the fluid environment around them as they're swimming or as they're feeding, which is an important metric for understanding their mechanics. And then finally, a fun outcome, but this is where we could really use a lot of help, is taking these light fields and constructing from the depth maps, the 3D reconstructions of the animal surfaces. And the reason why this is really, really important is that we can take these 3D reconstructions and bring them into computational fluid dynamics solvers like immersed boundary methods, for instance, to actually evaluate movement and how like energetics or efficiency changes as you vary some of these mechanics parameters. You can do that within a computational model. You can't do that in situ with real animals. And so what we're trying to do through this approach is to provide essentially labeled data, but validation data for these computational models so that mathematicians and physicists can then run with them and evaluate a variety of metrics around these organisms. So if you have any ideas, I went to a monocular depth estimation session earlier today, and with the ML tracking camera system, the stereo camera system, plus this kind of camera system, we would have essentially a labeled data set for monocular depth estimation, for instance. So things to think about, data we're already collecting for very, very different use cases, but could be useful, I think, to the community here. And then last but not least, and I'm going to blast through this because I want to leave some time for questions. Really what's starting to happen within our group is, while we started on using remotely operated vehicles primarily for all of our work, there is more and more of an interest for us to be doing these kinds of activities or work on autonomous systems for some very, very good reasons. So this is a plot that shows the state of long-term biological observations in the ocean. I think I use this paper in almost every presentation I give now. And the color indicates, in worldwide databases where you can access this data, regions where you have long-term biological observations. So, but it's also constrained to just near the ocean surface. And what the conclusions from this paper was that about 7% of the ocean surface area is covered by long-term biological observations. So only the surface of the ocean, and even that surface, we've only covered about 7% of it. And then the ocean comprises, 97% of the habitable ecosystem on our entire planet. So it's not a surface, it's a massive volume. And then what makes it even more challenging is about less than 1% of it has actually been visually explored, either by a human or by imaging system. So those numbers are awful. On top of that, it's estimated that anywhere from 30 to 60% of life in the ocean has yet to be described, AKA is unknown to science. So think about all of these unknowns, think about all of these gaps. How on earth are we able to achieve filling these gaps if we continue to use really expensive, often difficult to operate systems like the remotely operated vehicle I just showed you? What we need are autonomous systems doing essentially equivalent missions, similar to what we do on remotely operated vehicles, and then running those emissions to scale. Now, if we are successful at doing that through autonomous systems, all of a sudden we're collecting lots and lots of visual data. And I think this room can easily appreciate that. And we did a sampling of how much visual data is already available or exists in the community. So querying the US exploration-based institutions that generate visual data, there's more than 300,000 hours with less than 15% of it being processed, i.e. annotated. And then if you want to fill these gaps, you need to have an exponential increase in data collection. And so that means we're just facing a deluge of visual data. So how on earth are we going to solve that? Well, this is our first attempt. FathomNet is a data set that is really built to be a distributed data source where people from different parts of the world can contribute their image data or their expertise to help us label all of this information. You can check it out on fathomnet.org. Thanks to the fine-grained categorization and visualization workshop, this year is our first year to do a FathomNet competition with Kaggle, which I think the results were just presented today. So this is the first time we've done this, and we're looking forward to continue to do that because, as you can imagine, there's a lot of use cases and challenges with ocean imaging. Another thing I want to highlight is that we are doing a FathomNet mini workshop on Wednesday. So the first 50 people that comes to the Vancouver Aquarium will give you a ticket and also involve you in an activity to help us build the data set even more with animals that are at the Vancouver Aquarium. So please check that out. And then, as Sarah had mentioned, FathomNet was really just the beginning. As we talk to more and more users, people who create visual data, who need to process it for very, very real reasons, like biological monitoring is, as you saw in that paper, almost non-existent. What we've been focusing on is building out Ocean Vision AI with the goal, right, of building data pipelines and architecture for visual data coming in and annotated data coming out by using collaborative interactions between AI and humans. So as part of this approach, we've been funded by the NSF Convergence Accelerator. We're really focused on human AI interfaces that span the gamut between experts and enthusiasts, with creating 3 different software products that achieve this kind of interaction. The first is FathomNet, as I had mentioned, and thanks to the NSF funding, we've been able to really focus on what should be the community interactions to provide motivation or attribution for people to contribute to the database. The one in the middle really is called the portal, and this is where if you have visual data, really it's a digital asset management system that does not exist at a large scale in the ocean science community. And so that is also being built. And then finally, something I'm extremely excited about is a mobile video game that enthusiasts will be able to play and help label a lot of content. And so all of these things together are coming in to try and fill these gaps around human verification, which frankly is more of a challenge, and then hopefully help us really scale our observational capacity using imaging in the ocean. And so some quick highlights. So already we've used FathomNet data in a project called ML tracking. This is where we've trained some supervised models, incorporated them into our vehicle controllers. And here you can see a demonstration of it on our remotely operated vehicle mini ROV. And we're tracking, in this case, a solstice jellyfish, and we're able to ignore or robustly track it as other objects swim by. We've also been able to incorporate this into the same vehicle, but in the case of trying to search for particular objects we want to study. So you'll see in the top left the different modes the vehicle is in based on what the object detector is telling us. And so in this case, we're searching for giant larvation. Surprise, surprise. And then as we come up on the animal, again, this is like 400 meters deep. This is in the wild as it gets. You can see there's some proposals. If the object matches our search class, then you'll see us transition from acquire to track. And so there's so many things we would love to do with this, right? It's one thing to have supervised models running on your vehicle. We really want to look at, open world object detection. There's a whole session today on open world tracking. Like those are those types of approaches we would love to pursue using these systems that we already have in place. So what we're also doing is we're building imaging payloads for autonomous systems. So this is the long range AUV. This is the chitin imaging system. It's a 7-camera payload. So that will give us multi-view perspectives as we're conducting missions. And then the goal, right, is to really piece all of this together into what we're calling supervised autonomy. So if you have a submerged asset like an AUV or an autonomous system, and remember, the ocean is a GPS and communications constrained location. You can't just push data up into the cloud and have it process and then give you information or decisions that you can then make on that data later. So what we actually have to do is we have to create like a data relay. In this case, it's an autonomous surface vehicle that's tracking your submerged asset. And through acoustic comms, you can actually transfer information. You have to make decisions around what that information should be, because again, despite having comms, you're still looking at maybe a couple kilobits of data that can be transferred between these two systems. Then once you get it up to the relay, if you have an image that you really want someone to review, if it's an object that's novel that your object detectors haven't seen before, then that could be sent over satellite connection to a user on shore that then provides some sort of feedback. Is this something that we should be tracking, or is this something we should be avoiding and moving on? So then once you get that decision from a user, that goes back to the vehicle and the process then continues. And so it's this whole ecosystem that we have to create if we're really serious about pushing ocean exploration forward. And the group has already demonstrated bits pretty much all of the bits of this process. It's now a matter of getting this all to work together, but also to do it as effectively as possible. So if you have any ideas or suggestions, I would love to talk to you. And you can imagine we have a lot of things on our plate. And if you are interested, feel free to reach out to me or check out the lab. There's also a lot of different ways you can interact with us. MBARI has an undergrad intern program in the summer. We also have a competitive postdoc fellow program. And so, feel free to take a look at those things if that should interest you. So I'll take a question or two at any time.
So fascinating research. We know that the plastics are threatening life on the oceans. Is there a use case where you kind of study this also with this kind of approach?
Actually, yes. We've done, remember I mentioned that DPAV has a dye injector. And so a study that was done with collaborators at the Monterey Bay Aquarium, we showed that giant larvaceans will actually consume microplastics. And because they're so good at filtering particles and food around them, those mucus structures wind up being kind of aggregates of microplastics in the ocean. So we also find microplastics everywhere in the water column. It's not just a sea surface or a seafloor thing. So it's a big challenge. But thank you. Hi.
Great. In addition to all the video sensors you have there, do you have any acoustic?
We do. We have plenty.
What do with the acoustic data?
Well, that's a good question. So we have a number of acoustic payloads. For instance, we have an upward looking sonar that it's called DEMOS, that is constantly collecting data in the Monterey Bay area. I think it's at the Mars node, so about 600 meters depth. And so, that's tracking movement of vertically migrating animals, for instance. But we have payloads on our AUVs as well as our ROVs as well. Kelly Benoit Bird is actually the lead, I guess, scientist at MBARI focused on bioacoustics data. Way in the back.
Can they reproduce that mucus somehow synthetically? Can that be a way to filter microplastics more efficiently?
You're not the only one who's asked me that question. And I don't know it's possible. I mean, I think for me, at least what I'm really interested in from like a mechanics perspective is why do these animals have such complex structures? Is it a passive mechanism for particle selection, either by size or by content? I don't have the answer to that yet.
To the full autonomous systems. Things working against us is that economies of scale.
Yep.
Quite niche. How do we actually get there economically?
Yeah, there's a data problem, there's an access problem, there's a hardware problem. And really what we've pivoted to during the pandemic, et cetera, is focus on the data problem. That's why, FathomNet now exists. We're focused on that for Ocean Vision AI. But we're starting to see some movement in the low cost hardware space. There's a lot of people trying to build, low cost image sensors that are either drop cameras or can, have float capability so they could change their vertical position in the water column over time for low cost. I think one of the few groups that have managed this really, really well is the physical and chemical oceanography community. If you want to look up the biogeochemical Argo or the Argo float program, this is where you have these autonomous floats that just have a buoyancy engine that change the depth of their observations. And they have a pretty significant coverage, not through the major ocean basins, but also more recently through the Antarctic basin as well. So we essentially need to get to that point. But in order to do that, we really need to think about the algorithms we deploy how do we down sample the data we're collecting so that the useful information gets shared in these, you know, data and GPS constrained environments. Cool. Thank you, everyone.
I am, yes. Just need to plug that in. You can, put some on. I need to get my adapter. It's too heavy. This will never work. Now it is too heavy. Too heavy. Yeah. Okay.
So here and also here.
Yeah.
Okay.
Should work. Yes.
Yes.
Works. Oof. Sta quasi. Screen should be. Oof, worked.
Yes, good.
I just need to take this out. Oh. Good.
Davis? Yes. I have of the environmental computational science laboratory, CVPR, where he studies the Earth from above with machine learning and computer vision, and I think
Also from below now. I need to change that description.
in 2017 he became assistant professor.
Thank you, Sylvia. Thank you very much. Hello, everybody. How are you doing? Good? Okay. Very difficult to go after you. So I'll do what I can. So thanks for the nice introduction. And today I'm going to talk about something different. I mean, if you have heard me talking before, maybe it has happened. I usually talk about terrestrial problems with drones. And this is a new project that we are running since 1 year after, more or less. And I'm very excited. That's a new thing. So, I mean, indulge me if I say something stupid, but I do what I can not to. So the idea today is not only to show you beautiful pictures of coral reefs, but also to see what we can do to monitor that in a very special ecosystem, which is the Red Sea. So let's try to pass the slides. Yes. So again, we talk about life underwater, which is something new, at least for me, and in particular, so this is very relevant for Sustainable Development Goal 14. And among all things we can talk about life underwater, we'll talk about coral reefs in protection, restoration, and conservation. Right. So why this is important? Again, I might repeat something that has just been said, but the corals are not just pretty. I mean, they are, but they also are very, very important for our planets and our health for a number of reasons. So first of all, it's a source of food, directly or indirectly. So directly because fish the corals, and indirectly because we eat the fish. So since the fish is the main source of protein for many of us, protecting corals, it's a good thing to do to keep that chain untouched. Then we don't want the microplastic pollution. So also it is important regarding the fish. For the biodiversity, well, coral reefs are not very much. They do not cover very much of the oceans, but they host a quarter of marine life. So actually, it's pretty, for me, important that we make them survive, because it's the house of the fishes. And finally, it's not only about fishes, but it's also of the protection that corals give to the coastline, because they break the waves and they help us protecting from erosion. And then they're also beautiful and support tourism. So there is a lot of ecosystem services that corals give to us. And despite all this, well, we have basically lost half of the corals worldwide in 50 years. And okay, there's also problems of plastic pollution that are further exacerbate these problems. So the question that, of course, we are actually for animals here, and talking to an audience that it's probably more, which is easier to answer these questions, but I'm pretty sure we all agree that, you know, the future on the left is the one where we want to be, and not forcedly the one on the right. And you will be surprised, I mean, how far these two places are. They're not far at all. These two pictures were taken in a single dive. Okay. So basically, corals are under threat, okay? And they're under threat because of a number of reasons. So first of all, there is the water temperature increase, which is due to a number of factors, and they are quite heavily anthropogenic. And it's not on, and more on top of it, there is also all other kinds of stresses that can be the pollution of the water, or the exaggerated usages of some reefs for tourism. And I don't know if are you divers? Who's a diver in the room? Cool. Okay, half of it. Very good, very good. So when you dive, you don't touch them, right? But if you go to a very touristic place with coral reefs, you can tell me if people respect that very simple rule. Probably not. So all this together makes that, you know, beautiful places like this are already not so easy to find anymore. And if you continue like that, there probably won't be many left. So this is why, with some colleagues at the PFL and in other institutions, mostly in Europe and in Israel, we've tried to really concentrate on the Red Sea as an ecosystem to study. First of all, of course, there is a practical consideration. It was the closest, but it's not the main reason, obviously. It's because the Red Sea, it's a very unique ecosystem in terms of coral life, because our Israeli colleagues in Eilat have made this discovery, which was quite interesting, is that corals in the Red Sea resist to increasing water temperature much more than their cousins in Australia or wherever you look. So if corals in Australia bleach at 1.5 increase in temperature of water temperature, in the Red Sea, they resist up to 4 or 5 degrees. Which is, I mean, it's a significant extra resistance. So we're basically trying to find why is that, okay? And there is a number of hypotheses we have, of which one of the most promising is now mostly genetic and historical, due to the fact of the the Red Sea is a closed sea that has only one entrance, which is here at the bottom. And back in the ages, the Red Sea was lifted, and so it became a kind of a pond without any inflow of fresh water, which killed basically all life in the Red Sea. Then the land subdued again, and it was again filled by fresh water by the south, and it was recolonized by corals. But which makes that the corals had to migrate up north, and going through this very, very warm area here. And they had to adapt to this warm temperature, which makes that now we find corals that are very resistant to it, even in the north where the water temperature is much lower. So that's our main hypothesis. And we're trying to, confirm this. And basically we're going quite often actually to take samples and map the Red Sea ecosystem. So we have this transnational Red Sea Center, or PFL, which is supported by the Swiss government, where we have we're trying to basically creating this regional monitoring network of the corals together with all the countries that are in the regions, which we train by going there. We make seminar, we train divers that are from the region, and we try to set up a monitoring system where basically it's not always us going there and showing everybody how smart we are, but we're actually training people there and making sure that there is some capacity building, and then everybody is empowered and works together to this type of monitoring actions. And it's working pretty well. So we've been doing that for now a couple of years. So 1 year of substantial work, and there we are. So of course we are at CVPR Animals, we are at CVPR. So I will talk less about the diplomacy part of things and how you make different countries on the table for conservation. But I thought of talking a little bit more about our monitoring system, which, as you will see, I mean, it's extremely simple, and contrary to what has been maybe shown a second ago, which is fantastic. But I will explain why in a second. So we started with the idea that, I mean, we're not the first mapping and monitoring corals through imaging, but maybe one thing that this previous systems didn't have that we do have is that they were pretty time consuming. So here we are talking about a system that to map a 28-meter plot times 6 meters was taking more or less 21 hours to create a 3D model of this pretty tiny section of the reef, which was done in a fantastic quality and resolution, but it was difficult to scale that up. And then more recently, there are other systems that are very good when we go to the Bentley classes recognition, but then they're based on setups that are extremely expensive. And this is also something that we didn't want to go to, because first of all, you need a lot of money to build a very expensive system. And secondly, because I mean, our idea was to make monitoring that will be repeated and empowered by people that are on site and they cannot afford a $50,000 setup for a nice hyperspectral camera. So basically, we thought, okay, how can we democratize coral reef monitoring by having something that is scalable so that we can apply at several places with very cheap setups, so with very low entry prices of the system, and keep it simple. Okay, that was our bet. And the system we came up with is something based on some cameras that you can acquire on the market. So you go to your favorite electronics store and you will buy a GoPro camera. That is a little camera like this. We have a system. So the Mark I system was a okay, and I have to say it like a Barbie thing on plastic, and we were putting our cameras on it. Then Mark II actually was with a metal stick, and now on Mark III we are on several cameras mounted on the same. We're basically, filming the reef while diving. So there is a human operator, but basically the whole setup is less than $1,000. So it's something that people can acquire, and we can also leave to the local communities when we go to map in one place. We leave some cameras, we train people, and then they keep doing the monitoring. All right. So computer vision part. So basically, we have built a model that's starting from an ego motion video, so the video that is taken by the GoPro camera, performs 2 tasks. Okay, one task is the depth estimation, so the estimation of the depth of what we see, and the second one is the semantic classification of every single pixel in each frame of the video, and then we use them concurrently to construct a 3D model. So of course, one of the two tasks is supervised, the one at the bottom. So we have our students. The advantage in a university is that we pay our students. We pay our students. And sometimes our PhD students, hi Valentin, to make the the annotations for us, to creating this very well-performing semantic segmentation models. And the good thing is that again we try to keep it as simple as possible so that we can 3D reconstruct in video playing time. So it means that the model doesn't have to run for a million hours for one video. If the video is 30 seconds, in 30 seconds the 3D model is done. So that's we're almost there. We are 18 frames per second. At 24, we can say we are in plain video time. So very close. And we tested in 3 places already last year, in Israel, in Jordan, and Djibouti. Okay, so there are 2 parts. The first one is the depth estimation. Okay, so in the depth estimation, you need to do depth, you need 2 eyes, and so we do monocular because we have 1 camera. So basically, we play the trick of the learning depth estimation, and which means that we take 1 frame, the following frame, both times we estimate the depth. Then with the homography between the 2 poses that we estimate as well, we project 1 depth estimation into the other, and then we have some errors to backpropagate. Okay, we do it frame A to frame B and frame B to frame A, and then we can have concurrently the prediction of the pose and the prediction of the depth. And the encoder is based on ResNet if you're interested. And and not only we use when we encode for 1 frame, we not only use that specific frame, but we use a concatenation of 7 frames. So the one we are looking at, the 3 before and the 3 after, we concatenate them into a context encoding plus the encoding of each single frame, and then we use them all together for the 3 tasks. Okay, it doesn't change much, honestly, on the results to do this trick or not, but it doesn't add much to the computation either. So why not doing it? Okay, and let's hope that this goes, because Jonathan sent it to me during the night. Yes. So here you can see a little bit the results. So everything that is blue is close by, everything that is red is far away, and this is the result that it is calculated in video time by the algorithm. And here we are looking at the reef in Eilat, Israel, if you want to know exactly where we are. Okay, so this is the top part of the diagram. How am I doing time-wise? Okay. All right, and then this is the second part, which is a semantic segmentation pipeline. And here we use something that is basically off the shelf. So it's a ResNet backbone, and we use a Unix structure, so that at every single pixel we can do the predictions of the different classes that we are looking at. So the annotation looks a little bit like that, and these are the results of the neural network. So you see, we have a number of classes, and not all of them are Bentic classes. You see, there is, for example, a transdecline that you can see in green here. It's not something that we use to reconstruct the reef. But if we don't want to use it to reconstruct the reef, we need to detect it. And the same goes for the fishes, for example. So this is why we have all these strange classes. For example, the human class. So you can see here at Guilhem, our head of operations, who has very gently accepted to pose for us for this frame. And the story is that we are basically detecting human bodies and far away. Everything is far away, that you can see it in blue. It's not forcibly water, but it's something that is very far from the camera. And when we know where these pixels are, when we 3D reconstruct, we can exclude them from the reconstruction, because these are not part of the reef. But so without detecting them, it's difficult to do it. And so this means that we can go from a result that is like this. That is what we were getting at the beginning when we were doing that. We were doing distraction from motion without excluding this human, fish, transdecline pixel, which is a bit of a mess, because you're seeing a lot of parts of these divers in the reef. It's disgusting to think about. And this is the result when you don't do it. So when the person is still a person and not reprojected on the reef. you can still see some of this body even in the last results. And so in the end, all these systems used concurrently, and let's cross finger that this works. Yes, it looks like that. So these are the different parts of the model. So the one on the top right, you have already seen it. Is the depth estimation. Here we have the uncertainty estimation of the classifier, which tells us which parts are we sure or not. So, so it's a kind of a border detector and faraway object detector. And here is the semantic segmentation result. That, of course, we are not playing temporal consistency at all in the semantic segmentation network, mostly because it will add considerably to the forward pass computation time. So in a way, we try to keep it simple, even though the final result is not perfect. But just remember that we have then the 3D location on every one of these points, and then we can do majority votings on multiple frames. So which means that if you're making a mistake in a frame, it's not a big deal. Okay, so this is the type of result that we get at the end. So okay, the transit is pretty narrow because it's one GoPro camera and we dive in very close. But now we have this new system with 6 cameras, so it means that the footprint gets larger and larger because you have multiple cameras. We don't do stereo for the simple reason that the idea is to scale it up then any possible reef. So everybody who raised the hand before, if you have a GoPro, you will work for us, just that you know. And so if you go on holidays and you take a video of a certain reef, I mean, we will make a system where you can upload it, and this will like nourish our dream of a global coral reef map. And the cool thing is that you don't only use the semantic segmentation to filter out pixels on the 3D reconstruction. We also then get the semantic classification of the benthic classes for free, which basically allows us then to characterize every site with respect of the different composition. Some reefs are more sandy, more rocky. We can estimate how many of the corals are dead, and so on and so forth. I hope I'm on time. Yes, very good. Oof. Okay, so that's it. These are the wonderful persons I work with, and they go on expeditions with me. I mean, I hope I will join an expedition soon. It's planned. It's planned. Yeah, I mean, how to conclude? So I don't have to convince you here that the threats to oceans are real, and also that it's a large share of the biodiversity we have of the planet. So we need to care about the oceans too. And this is important also for us as individuals, because we rely a lot on what the ocean offers us. And there is a lot of data. We have seen it before, maybe a little bit more. And yeah, in this specific project, we are very happy because we're working with marine biologists and ecology persons and fantastic divers in the team. So if you have questions, here I am. That's everything I have for today.
I have a question, Devin.
Please.
It's really cool, and I love the focus on like keeping costs down and like scalability. Do you have a sense for like how good is good enough for these models? Like, what are they going to be used for downstream? Like, what are the important parts to monitor? Or understand how to understand genes?
I mean, it's not an easy question. It depends on the goal of each part of the study. So for what we need it, this is good enough, because we need to have a rough estimation of the composition that we want to have on the entirety of the Red Sea and then monitor it over time. So having a shift of a little something due to an error will not change this type of results. But it's also true that we go with colleagues that are geneticists, and they really want to see these genetic reasonings we had about the evolution of the corals of the Red Sea, and for them having the exact location of a part of coral that we have been sampling is very important. But I think the two objectives complement each other, and we could imagine having a system that is more, ad hoc for when we go with the geneticist that will map more precisely, and then having a system like this one that is coarse, but still allows you to cover like larger portions. Yes.
So the colors of objects, coral reefs and underwater change greatly based on the sun conditions, the depth of the water, light attenuation.
Yes.
Did you try to do any color correction or enhancement of the imagery?
Not yet.
No.
We are working on it. But there are some persons in the room that I think that have a lot of experience in that. Hi. We'll talk later. we are looking at it now to color constancy correction and things like that. Yes.
Basically, how do you get the location information for the measurement?
Yeah.
Or that is, you have GPS in the water.
It's I knew I didn't have to come after you. Sorry. All right. So how do we make sure that the transits are at the same place, basically? Okay, yeah. So for the moment, it's pretty rough in the sense that we go with the divers out. We put the line, and we have the beginning and the end of the line measured with the GPS, and so we try to follow a line that is as straight as possible. So as long as we see the line and we run the same transit, we're fine. But if you go back 6 months after, it's not going to be enough. So now we're looking at settings that that have like relays on locations. So, but we are purchasing it, and it will add up to the cost. So again, it will be a trade-off between what we do for our own development and cool research project and what anybody can use. Yeah.
Did you do some sort of landmarking or oral?
We would prefer not to, but yeah, we would prefer not to. But at the moment, we do that for the time we stay in one place. So we put these 2 marks at the beginning and at the end, and then there is a floater, and that floater we measure with the GPS coordinate, and that's for the moment what we do.
Thanks.
Yeah. Emma.
Related to that, actually, is it possible to get inspiration from people who do biologging? And so they measure the depth using the pressure, and that's how they connect the points. So it's like for animals that are tagged with like GPS or whatever.
All right.
But when they go deep,
Okay.
yeah, pressure sensor and depth sensor, which helps you to get data how deep you're going.
Okay.
Get next point when they come. So they interpolate that data.
Okay. Yeah.
So that data could be realistically available. I don't know resolution of that, but that's used for underwater tracking.
Sounds good. Yeah.
That-
So the question is if you can use a pressure sensor to improve the quality of location,
pressure and depth.
pressure and depth. The answer is I want to talk about it. Yeah. Very good, very good. Yeah, I mean, for now, the fact is that we went there and then we realized things and we tried to solve them. That's basically how it happened. Yes, please. It's so how long does it take to reconstruct a dive site? That's the question. It's on the video time. So you run the video, and at the end that you have been watching it, you have it. Plus a little bit because we are 18 frames per second. So it's not exactly video playing time.
Quite understand how you are estimating the depth of the pipe, which is relatively straight, and you said you will move in a straight line. Moving along the pipe.
Yeah.
2 cameras, they'll always be straight. and
You're never.
Straight.
Yeah, but you're never 100% straight when you're in water.
If it is straight, you can't estimate the depth.
I mean, you have the so if you dive in straight, you cannot estimate the depth. That's the question. I mean, you have the. I mean, it's depth through movement. It's monocular depth estimation. I mean, you can do it even if you go straight.
The pipe was straight, you're moving parallel to the pipe, and you said you move straight.
Yeah.
You won't know what depth the pipe is, right?
No, but it's relative depth. I mean, we're not saying this is 22 meters, 0.5. It's relative to the position of the camera.
Because there's no texture on the pipe, and you said you're moving in a straight line, and you see the same image throughout your journey. And
But not because everything is moving towards you. It's not the same image all the time. I mean, the coral landscape changes as you move.
just talking about the pipe, you said you need to detect the pipe.
Pipe? The line, it's just a string. we're trying to reconstruct everything else. The string is the only thing we don't want to reconstruct, actually. I mean, it still gets in because it's in the images, but we don't need it. We don't need it in the 3D model because it's not part of the reef. So we basically remove it. Yeah.
You said we need to detect it to remove it.
Yeah, by the classifier. Okay, now I get the question. Is the classifier that detects it? So we have a class line and we classify it. Yeah. Yeah. Sorry, I didn't get your question in the first place. Yeah. Yes.
So is this data public?
It will be. So is the data public? Yes, it's part of our engagement to make everything public. We're still processing it. Yeah. In principle, yes.
So now that you're on that slide, can you give an example of what class is segment there of the bottom left?
I knew. Yeah, I knew I should not have changed the video this morning because Jonathan prepared a prettier one. But now that I have done it, I will try to explain you a video that I've seen once this morning at 6. So the line is in the bright green. We have the massive corals here. The fishes should appear anytime soon. So you have the fishes there. Oh, it's difficult to do that in real time. So this is the sandy bottom. Let's see. So basically, we have the massive coral, the soft corals. We have them when they're alive, dead, and bleached. That's more or less the classes that we have. Yeah. So this is like classified as sand, for example. Yeah. But it's true that this video that I will try to stop now does not correspond to this color code because that was the old video and I just changed it this morning. Sorry about that. Yes.
Then do you have objects that are kind of man-made objects staying in water for a long time so that they are,
yeah,
an ImageNet would not detect them anymore, for instance?
Yeah. So the question is if there are objects that are deteriorated because they have been in water for a long time. Yeah, I mean, the model is fine-tuned with this type of categories. And as you can see, there is a category that I'm looking that's rubble. Yeah, rubble. So we have a category rubble and we learn from it using those frames where we see a lot of, unfortunately, plastic and things like that. So we train the model to recognize them.
So you could actually couple this with a, let's say, robot that will pick the rubble eventually.
That would be amazing if we could couple that. So the question is, could we couple that with a robot that will pick up the plastic? That would be super cool. Okay, thank you so much.
So then we went on to giraffes. I've seen an amazing amount of sea life, and now it's time to introduce to our scene horses and dogs. So it is a great pleasure to introduce Albert Ali Salah from Utrecht University. He has made numerous and really inspiring contributions in the field of human behavior, computational analysis of facial expressions, interactions between parents and children, doctors and patients. And he's also behind the newly established AI and animal welfare lab in Utrecht. And this will be the topic of today's talk. So welcome.
Thank you, Anna. Thank you. Okay. Is it visible also in the Zoom, do you think? Something weird?
Right. What can you stop sharing the
It's interesting.
restart sharing again?
Yes, I will try to do that. I need to go out
Yeah,
of this.
can you click that stop sharing?
I don't see it here, but yeah. Share.
Oh.
Didn't work. No, it
Oh.
doesn't work.
Why?
And show stop share.
Oh,
And
yeah.
now it's showing. Okay. So let me try to do this again, maybe. No,
No.
it's very weird.
Oh,
Okay.
yeah, maybe
So now you can stop the Zoom sharing
I could,
and you start the presentation first.
but then I will not be able to.
I think you can. So
Okay.
you can simply task switch. So command plus tab or something and Zoom.
And then start sharing.
Yeah. It's specifying the second monitor. Fantastic.
Okay, that works. Yes. So the title of the talk is completely a misnomer. It's not about AI and well-being in general. It's mostly about dogs and horses. But I want to talk a little bit about the AI and animal welfare lab that we have just announced a couple of weeks ago in Utrecht University. And then I will talk a little bit about pain estimation work that we did in Utrecht, and then conclude with some challenges that we're facing. So this welfare and AI and animal welfare lab is one of a series of AI labs recently established in our department, and these are interdisciplinary labs. And this one is mostly led by people from the veterinary science faculty. From our department, me and Remco Veldkamp are involved. We're both working on computer vision, multimedia, and multimodal interaction. But we have a bunch of veterinary scientists, but also animal ethologists in the lab. And some of them are pretty big groups with a lot of history of research in these areas. And the aim of the lab is basically to create some animal-centric artificial intelligence empowered models and applications. It's all very hands-on, very practical, and very much directed towards the goals of the veterinary faculty. There are a couple of thematic focuses. I'm presenting you this because I'm hoping maybe there will be some future collaborations between our lab and with people in this room. We're already discussing with the University of Haifa to start an ITN maybe on educating the people that have both the veterinary and computational skills to work in these areas. And the thematic focus that we are working on more is locomotion and activity detection and recognition, automatic monitoring of welfare, and human-animal interaction. The horse research that kind of got me into this is actually pretty old at Utrecht University. The first veterinary faculty was established in 1762 in France, but very shortly afterwards, in 1821, there was one established in Utrecht, which became part of the university in 1925. So the horse research is quite old at the university. And over time, it has also developed into certain focus areas, one of which being locomotion and lameness detection. So I'll just show you a bunch of the projects that are being conducted under the aegis of the lab now, like the EquiPro equine gait analysis project, which is in collaboration with a startup a spin-off from the university, working with wearable sensors. And one of the things that that make wearable sensors suitable for this kind of work is that the sampling rate is pretty high. So this is 1,000 hertz sampling rate sensor, which is something, you can detect subtle patterns that are not visible to a human eye with this kind of motion detectors. And the group has recently also organized the Canine and Equine Locomotion Conference in Utrecht a couple of months ago. Another project that involves computer vision is 3D printing of hooves for horses. These are things that you do not need to nail into the horse's hoof so that there is less risk of laming the horse. And then you can adjust these hooves to the individual needs of the horse at the millimeter scale. The problem is, of course, you have to streamline this so that it can be quickly done by, let's say, a horse owner or a veterinary, or and then there are some issues that needs to be solved because you're producing these horses' hooves from plastics, and every time the horse is going around, you're releasing a little bit of plastic into the environment. So we should think about using some degradable materials in this kind of thing. Improving hen welfare through computer vision. if you have a hen coop, you have many of these animals, so it becomes very difficult to tag them with wearable tags. So we need to rely on computer vision to be able to pick up the sick animals, for example, from their behavior. At the moment, the computer vision algorithms do not scale up very well for this task, or they will require a lot of computing power to individually track these animals. So there is research conducted to improve this. Another project, this one is a Horizon project, a European project called A-Wish. And this is for improving the welfare. Well, it's a bit strange when we talk about welfare in the slaughterhouse, but it's an automatic monitoring of the slaughterhouse, the complete pipeline, basically, on the farm, unloading, transporting, and slaughter. So one part of the lab is also working with companion animals, dogs and cats. And this is research by Saskia Arndt, who is an ethologist, and they monitor nocturnal activities, and they do sleep assessment for dogs. And if you have ever worked in sleep assessment, it's a very difficult problem. It's a challenging problem if you want to do it remotely. Even in humans, it's pretty difficult. But they do, they use some wearables plus the video observation, and they create these ethograms, and they try to describe the sleep activity of dogs. Two projects that I'm going to talk a little bit more about. One of them is pain estimation in equine faces, and that includes horses, donkeys, and mules, maybe. And then the second one is dog pain estimation for camera. So this is a video-based analysis project. Just a few days ago, accepted for presentation in the Affective Computing and Intelligent Interaction Conference. It's going to be in the coming months. So for pain estimation in equine faces, our aim is to understand pain indicators. And then there are a number of grimace scales developed for assessing horse pain from the facial expressions. One of these scales was developed at Utrecht University Veterinary Faculty, and another one was developed in Sweden. And we use both of these. We use actually multiple scales, because in this task, we are trying to do this from images, which is actually quite difficult. But we created a database and then annotations, and we implemented a computer vision pipeline. The two scales that we have used, you can see the references, one from Thijs van Loon and the other one from Dalla Costa. These grimace scales actually include a lot of indicators that are based on video. So, for example, some of the indicators require that you listen to the sound the horse is making. For example, the flagman behavior, this snorting or head movements. So they're not possible to estimate from images. What we did was we selected the static features from both of these scales and combined them into a grimace scale, but that is based on images. And so you can see here the 6 areas on the face of the horse that we're looking at: the ears, the orbital tightening, the eyelid, sclera, corners of the mouth, and the nostrils. And each of these things, these locations, are scored from 0, which represents no pain, to 2, which presents severe pain. So the veterinarians, when they scale the pain, they look at these indicators and do this per area. And then you sum this up to kind of get a score for the entire pain. We collected 3 different data sources we used in collecting our data sets. The horses from the Netherlands is images provided by horse owners over all the country. And then some of them have bridles, some of them have not. So there are quite a bit of variation in these images. Then we have a small data set that is horses with clinically induced injuries. That's an clinically, ethically approved procedure for inducing a short amount of pain for a certain duration. And then we have images of donkeys from a donkey sanctuary in the UK. We wanted to see whether the models that we developed for horses can we use them for other equines as well? Can we use them, for example, for donkeys? And here you see some of the landmarking tasks that were implemented in these frontal, tilted, and profile faces. Now, I did my PhD on 3D face recognition, and I worked a lot on 3D facial landmarking, spent a few weeks landmarking the FRGC dataset that came out when I was doing my PhD. Now, facial landmarking in horses is much more difficult because in one single pose, you cannot really capture the entire face. So it's difficult to use the contextual information. And then a single model, if you try to build for all the landmarking tasks, then it will be too complex. So our solution was to build landmarking modules that are specific for poses. So we built actually 3 of them. And then there is less public data for training. I mean, for human faces, what we do is we, download the IMDb, and then you have a lot of human faces with names, and then you can train a neural network for identification. And then you can strip the last layer and use this for transfer learning, because now it has good internal representations for what the face is doing or how it appears. But we don't have this kind of data available for horses or donkeys. So that's basically data scarcity problem. The horse code obviously has a lot of variations. That's also a challenge compared to humans. But when we came to dogs, that was much, much worse. But still, we have different colored horses, and that's still a challenge. And the bridle will create some occlusion problems. Now, in humans, typically when we do facial landmarking, we normalize by interocular distance to quantify the error. So if, for example, if your detected landmark is 10% of the interocular distance away from the ground truth, you say, okay, that's close enough, we accept it as true. And then when it's further away, you say that's incorrect. But in the horse, you rarely have both eyes visible in one image. So we selected the eye nostril distance as a normalization factor, and we basically created systems measuring the error as a percentage of this distance. Now, the pain score distribution in the data set is quite uneven. So you can see the 6 locations that we're looking at, and the light blue is 0 pain. And you can see that we have very few instances with severe pain included in the data set. And that, of course, creates another challenge. One thing we tried to do to improve the landmarking is to use data augmentation. And what we did was we took a 3D horse model, and then for a given horse that we have in our database, we thought if we could align it to the 3D horse model, like do lifting to generate a 3D version of it, then we could image this 3D horse from different locations and then generate synthetic images. Now, this is actually inspired by the charades dataset. If you know that, this is a work that does this for human poses. So they use a human pose generator, and then they just paste the human on these different backgrounds, and you have these floating humans in the ether. But it still improves pose estimation greatly. But in our case, it did not work that well because we had only a single and a very simplistic horse model. Now, among the posters today, there was the bite, which is very promising for us. So I have one student waiting for me in Utrecht who is going to start a thesis on applying bite to improve the data augmentation with dogs. So maybe with improved 3D synthesis models, we can solve some of this problem. For this one, we used Levenberg-Marquardt optimizer to kind of align the 2D image, followed by a thin plate spline model to warp the 3D model to the 2D image. And then we used the symmetry to fill the pixels that we can't see in the 2D. For facial landmarking, for those of you who are familiar with human face landmarking, we used the ensemble of regression trees and supervised descent model of Fernando de la Torre in combination. And then we try to focus on the stable landmarks, because when the pose changes, the contours of the face, they will move around the face. So they are not very good in terms of automatic estimation. It's, you know, it's a rather safer bet to focus on the stable landmarks. So that's the whole pipeline. Then we have first of all a pose estimator, and we use a model that was initially trained in Cambridge for sheep pose estimation. Marwa Mahmoud's group. I think some of you already know this, familiar with this work. And then we have pose-informed landmark detection. So for every pose, we have a ensemble of regression trees. Then we use, because we lack the data for end-to-end deep learning, we use a more classical computer vision pipeline using hog features and a support vector machine classifier. Now, if you look at this, so these graphs show you the inter-rater agreements for 3 human annotators for no pain, moderate pain, and severe pain cases, and for the 6 different areas. And you can see here that for moderate and severe pain, the human annotators do not agree with each other very much. So our ground truth annotations for severe pain are not very strong. That's a bit of an issue, obviously. But apart from that, so our F1 scores are roughly changing from 0.5 to 0.8, depending on where, in which part of the face you're focusing on. We're getting decent results. So pose detection from the horse's faces, that works pretty well. 3D-based data augmentation does not work very well, does not give us the boost that we're hoping at the moment, but there is room for improvement. We're going to keep working on this. Variations in head morphology between the different species and different breeds of horses actually make generalization quite difficult. We tried transferring to donkeys. It did not work very well. So for donkeys, you need to basically train new models with ground truth annotated on donkey faces, was our conclusion. And then we had very few samples for the severe pain class. And it's very difficult also for our veterinarian colleagues to collect data with pain. It's the procedures for inducing pain are rarely approved by the ethics committees. And then you need to just look at the animals that come with pain to the clinic, you know, undergoing certain procedures. And then you have some limited data set. The second project that I want to talk about is dog pain. That's a collaboration with a veterinary faculty in Turkey, in Ankara University. And they have collected data from dogs that came to the clinic with with under certain painful situations. So this is naturally occurring pain, not induced pain, but it's also very limited and difficult in that sense. And the aim is to enable video-based pain estimation. And for that, again, the data set is available from the veterinarians. If you contact us, there is an ethical approval procedure that you need to undergo before you can obtain the database, but it's available. For this, we combine several off-the-shelf and in-house tools. And the data set that we have, Ankara University Dog Behavior Dataset, has 61 videos collected from different dogs from a duration of 6 hours and 45 minutes, roughly balanced in terms of pain versus no pain. And then we have orthopedic or neurological pain, but we do not have all types of pain in the data set. The annotation is done by a veterinary expert, and you can see the distribution statistics. This is how the data looks like. So all the dogs are recorded in this little room. Most of the time, you do not have anybody present. And the dog's behavior, well, you see that the camera is not very ideal for this kind of settings. And this is something we have also seen a lot with doing work with human subjects. the clinicians would collect data for their annotation purposes, and then the illumination conditions will not be ideal for doing computer vision. We use a YOLO model to find the coarse localization, and then we use HRNet trained on animal pose dataset for a more precise pose estimation of the animal. And then we get these key points, which we interpolate via symmetry and from basically a bunch of simple rules, taking into account detections in the neighboring frames in the video. And then we normalize the pose. Basically, we take the dog to a uniform coordinate system, on which we have now the tracking problem. Of course, is a much more simpler tracking problem compared to the birds that you have shown us in the morning. But it's still challenging for us because we have very sparse data and very sparse annotations. We use two-stream network. We use a simple LSTM based processing the key points, and then the color images, we use a convolutional LSTM to process them. And then we stack these two feature vectors, and we have some attention layers to combine them efficiently. For baseline models, we use a grid search. We look at a GradCam activation to see what is the model basically paying attention to at the end. And then we have an F1 scores ranging from, F1 scores ranging from 55 to 74, but we can improve these with this proposed two-stream model to go up to 77% accuracy. There is room for improvement, obviously. The dog breeds are have a lot of variations, but this is the first work ever on pose-based dog pain estimation. So the next step is perhaps to combine this with the face of the dog and use face indicators of pain. The multimodal cues, for example, the vocalizations, attention to wound mobility, responses to touch, these are all things we may want to add in a multimodal framework. We looked at the limited number of pain expressions, and then we were very careful not to fine-tune the models a lot and not to use strong, deep learning models. Because of the scarcity of data, this is prone to overlearning. So it's very easy to, go up to high accuracies with a very complex model. We didn't want to do that. Okay, the future outlook. Well, in the lab, we want to focus on a bunch of problems ranging from sensing animals, especially pain, activity levels, medical conditions, and sleep monitoring. The applications that we have a lot of commercial partners working with us. So we're looking into training and interaction programs, home care applications, post-surgery assistance, welfare monitoring in different specific contexts. And then we need to look into the ethical aspects of all of these applications. This is my last slide. And I tried to kind of list some of the challenges and opportunities that perhaps you may want to discuss in this environment. There are some new data sets that include a lot of animals. They are sometimes not collected for the purposes of animal well-being assessment, like LION, but they do contain animal images, and they are a big help for creating key pose detection or segmentation models. But large and clinically annotated data sets, these are missing. So we need definitely this kind of data sets. Clinical validation for computer-based systems is necessary. What we notice is that in the clinical domains, in both medicine and in veterinary science, the validation is much more meticulous. Whereas in our domain, in computer vision, we just take a database, we say, okay, here is the ground truth, and then we start reporting accuracies, which is not enough. You need to really understand where the errors are coming from, and you really need to look into the individual cases. And also in psychology, this is a problem If you look at the texts of the old Gestalt psychologists, they would talk about each of their patients in great detail, because those are the insights that you want. We have this tendency to reduce things in our field, and that could be harmful in the long run. For computer-based analysis tools, we need simple, intuitive, and useful interfaces. That means this is also an HCI problem. So we need to work with the HCI people to create interfaces into the tools that we're developing. AI ethics research that mostly focuses on humans. The implications of these technologies for animals have not been discussed at all. I mean, if you look at the AI Act, there are very few implications for animals. And I think as a community, we should start maybe this, and we should press for doing research on AI ethics for animals as well. Regulations should be introduced and guided for healthy development of AI-based animal applications. And then privacy issues needs to be tackled. For example, if you're working with horses, sometimes you deal with race horses, and privacy becomes an issue because there are huge financial implications of, if you show this horse in pain or indications of lameness, these can have consequences. And then we feel that experts should be educated with interdisciplinary skill sets to develop animal well-being applications. And I've been talking to some of you, and what kind of skills would a PhD working in this area need? That's definitely interdisciplinary. There needs to be some animal ethology, some computer science skills, data science skills, but also statistics and other skills. So we need to develop programs for that. This is my email address. If you have any questions or comments, they are most welcome. And thank you very much. Yes, please.
Yeah, you mentioned that there were clinically approved ethical production methods. What would be considered an ethical method of reducing pain?
Yes, that's a good question. Sorry?
Can you repeat the question?
Yes, sure. So the question is, how can you ethically induce pain, right? So, for example, in humans, the cold pressor pain is an example, or plunging the hand into cold water to give you a sharp pain that is essentially harmless is one way. But in these horses, I think they're injecting something in the joints that induces temporary lameness. So I'm not a veterinary scientist, so I find this a justified question, but I think, I know that there are no consequences, like any long-term consequences to the horse's well-being, but the short period in which the horse is experiencing pain is there. So that's a valid question.
Under the hoof, like behind the stone, if it discomforts than pain.
Yeah. So yeah, I was shocked when I first saw that the iron hoofs are actually nailed into the horse's feet. So you might say, okay, is this ethical really? But it actually protects the horse from the uneven surfaces, and I think the way they do it is they don't hit any nerves, so that it's not painful. But yeah. Yes, please. The which one? We don't use it, but some of the models that we use are trained with data from that. Yeah. We use Stanford dog and ticks, but I think there's again, we manually annotated some of these, but we need more data basically. So we are going to also look at this data augmentation problem. I don't know it. So is it a recent data set or? Okay. Okay, thank you. I will check that out. Animal Kingdom. Yes.
did I understand correctly for the blockchain project, it was in virtual images?
For the dog pain project, we no, the whole video is annotated. So the veterinarians annotated the videos in the case of the dogs. So we do not have frame level annotations of pain. We have video level annotations. Yes.
Video annotations for horses as well.
Not yet. So one of the things is that we're lacking videos from horses with pain. But one of the things we want to consider for the future is also to introduce a rig for recording the horses that are brought to the veterinary clinic with multiple cameras, so that we can also have a 3D reconstruction. So 4D scan of a horse in pain would, I think, be an important contribution, but that requires a special setup, basically.
Yeah, it looks like you really need 3D or chip way. Don't have a lot of data,
Yes.
like in the case of the horses. And you need to have a specialized model for each pose of the head when you put factor out the pose.
Yes.
That I think would be beneficial for these cases where you don't have a lot of.
Yeah, Sylvia's comment is that we need 3D models that work well with because of the data scarcity. If we can lift a 2D image into a 3D model properly and then generate multiple images from that, from different perspectives under different illuminations conditions. So this is something, by the way, we also faced with human faces, right? Some of the signals are very subtle. So the noise level is actually sometimes larger than the signal that you're looking for. And that makes the problem very challenging. For example, if you want to detect sarcasm in a smile, right? So very small things, sometimes the timing of it is slightly off, and that makes it into sarcasm. You wait a little bit before answering. So it's also interaction. Sarcastic horses would be. We would go to cats if we want to detect sarcasm. Thank you very much.
Yeah.
Yeah. Did a-
Okay, I should be muted or not muted?
Keep clicking.
Okay. Can I connect? Yes.
Screen sharing.
Yes, I can totally do that. Okay. Should do my video too. I'll do my video too. Share screen here. That, and then have a slideshow. Close this. Go.
Yeah.
Can I go? Oh, I have to be on. Right. Here we go. Okay.
Yeah.
Okay, great.
Can you simply click the stay button?
Yes. Stay participant.
Oh, yes, good.
Okay.
The first oral is from Duke Myers, work automatic honeybee.
Awesome. Hi everyone. My name is Luke. I'm gonna be talking today. Let me pull up my notes. I'm gonna be talking today about the research that we've been doing towards automating re-identification in behavioral choice assays that we run with honeybees. This work was conducted at the University of Puerto Rico, Rio Piedras. with a collaboration called Deep Pollinator that includes a bee ecologist, an animal behaviorist, and a machine learning specialist. And we're all about developing tools for automated pollinator monitoring. So, automatic honeybee flower patch assays. What are those? Ooh. Okay, great. So, as you can see on the left, we have these two little plexiglass flowers that the bees are trained to come visit, and they will drink nectar from the center well. And these assays are used to assess decision making for the bees. And we want to be able to link behavioral traits that we see in these assays with genetics. And to do that, we have to scale them up massively. So we're working on automating the data collection for those. So our data collection system, as you can see on the right, we're videoing these assays from above as the bees come and visit these flowers. And our future overall pipeline is to collect video in the field, detect and track the bees in the frame, and then be able to link behavior observations that we make to the individual level. And that requires a re-identification system. So that's what I'm going to be presenting on today. So the video, here's a sample on the left. And we performed offline pose estimation to detect the bees and track the bees. And then we used that pose estimation to extract standardized images to use for our system. When the bees come and visit, they get a little drop of paint from researchers, 1 or 2 drops of enamel paint. And these compensated images centered on the waist key point were used to create a data set of 4,000 images spread across 27 identities. So that's 27 real world bees with their unique paint code. And you can see here kind of a spread of what some of those look like. And we also extracted uncompensated images, as you can see in the top right. And those will come in later, but these are both like part of our overall data set. So because we knew that we wanted to automate these behavioral experiments and have a model that reliably could re-ID new identities as we run these experiments over and over, we treated this as a contrastive learning problem, specifically focusing at the open setting as our really target use case, where the model is re-identifying individuals that it hasn't seen before. So we used contrastive learning with triplet loss to learn a feature vector for each individual, and then be able to re-ID between photos by using the nearest neighbor when projected into the feature space. And then we did this evaluation using both the closed setting and the open setting. So the closed setting is where the model was trained on all the possible identities that it might be evaluated on. But then the open setting, a certain subset of the identities are reserved to evaluate how the model can re-ID new individuals. And that's further split into the reference and query set. And so this evaluation was performed using galleries of 10 images. You can see in the top right where the particular images are sampled from in each of the settings. But basically, there's an anchor image that is selected from the test set, and then 1 correct image and 9 distractor images. And these are all embedded into the feature space. And then the nearest neighbor is selected to see if it matches the correct class or has the correct identity. In this particular example, you can see that it was able to correctly re-ID this individual, even though the background changed, the pose changed. And so that's kind of where we're trying to create a robust model. And then this example uses full body crops, but we also calculated the top 1 accuracy for 10,000 of these 10 image galleries for several different use cases. So for full body crops, but also just the abdomen, the thorax, and then the uncompensated images that I had talked about earlier. And so this top one performance is plotted here. Closed setting on the left, open setting on the right. You can see these two orange bars show the closed setting performance at top one re-identification for the thorax and full body crops at 0.99. So this shows that we have excellent re-identification in the closed setting using these paint code markers when the model has been trained on all the IDs that it might see. So you could consider this like a more typical classification problem. But we also see surprisingly good performance in just the abdomen crop. And that's really cool because it shows us that in the future, we could potentially use markerless re-identification. I mean, that's definitely like a direction we want to move towards. In the open setting, the thorax still performs the best of all the crops. But you can also see that it performs significantly different than the abdomen and the full body unaligned. And that shows us that in the open setting, the addition of the paint code and the alignment of the images significantly increases the ability for the model to perform correct re-identification. And that makes sense. It's easy to pick out a bee when it has a bright red dot on its back. But it's a really interesting way to kind of bridge the gap between traditional modeling, traditional marking with like tags, April tags, that can be really intensive and hard to do. And then more lightweight methods or even markerless re-identification. So these paint codes, we're presenting a new data set with thorax paint codes for honeybee re-identification. Using contrastive learning, we showed excellent performance in the closed setting. And our next steps are to gather more data to improve generalization in the open setting and also just to further evaluate the performance of this approach. On the right, you can see juvenile bees from days 1 to day 3 can't fly and they can't sting. So you can just grab them by the handful and paint as many as you can grab. So that's what we're working on this summer is getting more paint codes to use and then integrating it into our future real-time pipeline that can be deployed in the field to create these systems for automated pollinator behavior analysis at the individual level to test all sorts of things for the behavioral scientists that we work with. Thanks.
Background and like fake flowers.
Yeah. So the question, totally. So the question is, will we change the background? So we've looked at a lot of data augmentation techniques. These images, I'll go to the data set picture. They're actually 200 by 150 pixels. So pretty small as it goes. And you get a lot of bleed from the background. So even when we do background subtraction, it still really affects the like color makeup of the image. So like the blue and the white are kind of strange, but they're like neutral to the bee. Like the bee doesn't have a preference for them. So for now we're doing this, but when we like the circle image I showed at the end, they're doing that with a bunch of different backgrounds to test how that affects the paint code reading. Does that answer your question?
Did you try or have a look at existing markerless methods like T-Rex or something, which is showing hundreds of fishes identification?
Yeah, so we've done some work with markerless identification, particularly when it comes to the abdomens, because the abdomens do contain features that could be used. But for this particular data set, we haven't looked into it just because of the paint codes, and they need to be monitored by researchers anyways, because they capture them at a certain point. So like full complete markerless is definitely like a future step, but not as relevant as it may seem for this current system. Also, the re-identification using markerless and semi-supervised methods that performed really well for us required a lot more images and a lot higher resolution images. So that's why like we've been working on this approach currently.
Just a small follow-up. So are the color markings also have to be unique? So you have to generate some combinations for people?
So so we've been looking at that. So yellow, we have like several different bees that just got a yellow dot. And you can, like a human looking at it, can use the shape of the dot to distinguish it. And while like our model can do that as well, it takes a lot more potential input to get like all the possible shapes. So when we agglomerate all the classes into just all yellow as one class, we actually see really good performance in the open setting. So that's one of our other next steps, is like refining our paint code like choices to really match and be more distinguishable in the feature space.
So like, I was just imagining like a big B column. So right now you have 27 identities. So like for future work, kind of what kind of number are you trying to reach for IDs? So like, is 27 enough, or like you really need 500?
We're gonna need a lot. So like this work is sponsored by the USDA, because this automated pollinator like monitoring can eventually be used to like maybe select for traits that we're interested in, or look at like large scale genetic monitoring. So when you think of a hive, social insects, the whole hive has the same genotype. So like we eventually want to get to a level where we can set out an experiment and then have like a whole hive go through and just like run it over a course of days. So that's like our very far in the future. But we have some promising results on real-time detection and tracking. And so we're coming towards a real-time prototype pipeline. That's pretty exciting. And it's just integrating that into a field system. Yeah.
But the data set, does the data set be generating in the same days or cover a large period?
It's
actually working, or they are isolated?
So it's across 4 different days in the summer. 4 different days, and they're actually like several weeks apart. And then it's like several videos per day. Any last questions? Yeah.
Question about your answer. You said you wanted to focus with maybe the color and more colors, but you should just show that actually abdomen, just using the abdomen, you are able to do classification. I want to just focus on that, and like that you sort of make sure that you can cover so many individuals, and then you can cover eventually 1,000 individuals just using the abdomen.
So what we see right now is that the like lightweight marking methods perform best compared to the abdomen in the open setting. And that's our eventual target case, is to have a model that we don't need to retrain each time we move to a new hive to do this type of evaluation. And so that's kind of our target, and why we're working on more lightweight tagging methods rather than complete investing time in markerless. Thank you guys. Do I have to stop sharing? Oh yeah. Me.
All right, okay. Thank you everyone for staying back for the last talk of the day. So my name is Anuj Karpatte. I'm an associate professor in the Department of Computer Science at Virginia Tech. And my student, Meda Sani, who's the lead author on this work, could not travel. So I'm presenting for her. She's also online, so she can correct me if I make any mistake today. This work is in collaboration with Dr. Bahari Beckham's group from the Department of Mechanical Engineering. This is the Micron base group at Virginia Tech. And what we are doing in this work is very much related to computer vision for animals, but it's sort of different from the other types of animals that we have seen in this workshop. These are microscopic animals, bacteria, that are moving in highly dense backgrounds, hydrogel backgrounds. So the texture The textured background that you see here is the hydrogel. And these objects that are tumbling around, for example, over here, sometimes they appear very prominently. They are the bacteria that we want to track. Why do we want to do this? Well, studying and understanding the mobility patterns of bacteria is useful in a number of ecological and biomedical applications that involve bacteria motion. For example, in targeted immunotherapy, we are interested in loading these bacteria with drugs and seeing if they can tear through tumor cell matrices. And we want to understand what changes in the background, what changes in the pH or other conditions can change their mobility patterns. So if you look at what has been done in this area so far, there are two basic types of works that have been done. The first is where we would do fluorescent tagging of the bacteria so that they are nicely separable from the background that appears black. And this is, of course, easier to perform, but it has its own challenges. First, fluorescent tagging alters the physiology of the bacteria. And second, this cannot be done at the desired frame rates at which we want to study bacteria motion. There is also work that work with bright field images without using fluorescent tagging, but they have all been using liquid medium because it's easy to separate bacteria from the background. But liquid medium is not an accurate representation of the environments that bacteria observe or encounter in natural habitats. For example, inside our human body, which is mostly composed of extracellular matrices of cellulose and collagen and other such matrices. So in this work, we want to study and track bacteria movements in dense hydrogel backgrounds, for example, in agar or in collagen. And you can see that over here, the background is much more dense and it has a lot of texture features. So why is this problem challenging? The first challenge that we encounter here is how difficult it is to separate the background from the objects. Here is an example in agar medium. Right? We have two patches that we have zoomed in, and you can see that for this particular yellow patch where you see the bacteria, you have a nice signature that the bacteria is showing up as a prototype, which seems similar to the feature that you would observe in a background patch as well. And this problem is further exacerbated when you go to collagen, which is an even denser background, right? Here, the bacteria does not even have a typical signature, a typical prototype, and it's very much similar to any other background patch. This is strikingly different from many other conventional applications of computer vision. For example, when you're tracking pedestrians and cars, where the background is easier to distinguish from the objects. The second challenge is that we are recording these movements using 2D microscopy, whereas these objects live in a 3D world, and bacteria can go in and out of plane, right, in a translucent background medium. So what can happen is that a bacteria that was showing up prominently, right, perhaps like a cylinder or like a circle, may become blurred eventually in different frames, and it may again show up sharply in a different frame. This is what we are categorizing as easy, hard, and very hard. And so for every bacteria, we are annotating the bacteria into these classes based on the sharpness of the image or the sharpness of the bacteria. So this is another challenge, which is because of the imaging modality we have. The third challenge is the erratic behavior of bacteria motion. So these are generally modeled as random walk, and this is not as well behaved as the motion of pedestrians and cars. This makes it difficult to extract motion-based features to be used for tracking them. So to address these challenges, we have developed a very simple framework, a proof of concept framework, which we are referring to as MMET. And there are 4 modules to this framework. The first module is trying to augment the features that help in detecting the bacteria. So we are looking at motion-based features for every frame. For example, we are doing consecutive optical flow, and we are looking at the difference from the median background as additional features. Once we have these additional features, we are sending these inputs into 3 different models that are capturing 3 different types of bacteria modalities. We have the easy, medium, hard, defined according to the blurriness or out of focus feature of the different bacteria. We are using simple retina net models for each of the 3 categories. We're combining their predictions, tuning out false positives using a number of post-processing steps. And finally, we are applying the sort interpolation algorithm to track them. So that's a very simple approach. And in our results, we can see that even this very simple approach is able to achieve good performance in terms of precision and recall. Especially on the agar medium, we are able to achieve a performance of 0.82, 0.94 after tracking. And this plot essentially shows how the precision and recall changes with every step in our pipeline. For collagen, our recall is good, but our precision is still 0.51, and that's something that we are working on. Collagen is a difficult background to work with. So here is just 2 videos to show the scale of complexity that we are working with. On the left, you see agar, and the yellow shows our predictions, red shows ground truth, and you can see that it's doing a reasonable job. But if you go to collagen, right, you see these yellow false positives, right over here, which are very similar to some of the false negatives if they show up somewhere. For example, over here, it's the amount of visual information that is contained here is occluded with a lot of noise. So this is a work in progress. We are looking at other approaches, building upon this simple first cut pipeline, looking at deep learning based tracking methods instead of sort. And finally, we are using all of the work that we have done in tracking to go for forecasting or predicting the trajectory of bacteria for different strains with different genetic modifications and so on. With that, I'll like to please. Which figure do you want to?
So on the left.
Right. Yeah. The white. Right. So the question is, what do we see here as the white circles around the bacteria, right? And if you look closely, sometimes you have some really large white circles if the contrast is showing up well. Those are actually diffusion that is happening. And I'm not an expert in how these images are taken, but this is happening because the bacteria is moving in and out of plane. So when you have a very sharp looking bacteria, maybe in some of the frames you would see like a cylinder type of object or an ellipsoid type of an object. That is when the bacteria is in the right frame. But if it goes in and out of focus, you have these halo effects around it.
And about the data, I mean, you made a pretty good job at showing us how hard it is. But then I feel it's so hard that can you really get investors to ground truth?
Yeah, that's a very good question. So can we even rely on these ground truth annotations? Is there sufficient information in the data that the model can use? Well, that's a question that we are exploring. Our human annotator, which are PhD students working in Dr. Beckham's lab, they're able to identify these bacteria and they have the expertise. They have the training to identify them. We are trying to do some experiments where we can check with multiple students if they are able to find the same bacteria or not. But certainly there is some information that they are picking up, and we would like to see if a machine learning model can do the same job.
Let's do different kind of video acquisition, like light field LSR here, and light sheet microscopy to get like volume for this bacteria.
Right. That's a very good question. So I'm not an expert on the imaging, but I can certainly follow up on those directions. We are looking at some directions on changing the z-axis, but it's hard to find the right z-axis that works best for all the bacteria, because these bacteria are not synchronized, right? They are moving in all possible directions. And if we could capture all the z directions at one time, that would be great. As far as I understand, that's the limitation of the imaging that we are doing at the high frame rates that we desire. But I'm certainly happy to learn more about other imaging techniques. Thank you.
Yeah, the head of the.
So Kostas and Albert, I welcome you to the panel.
I think you can discuss with them.
And also joining now.
Check the.
Yes, probably.
Or put something in front of it because otherwise.
Yeah.
Do we have
let's do it.
or
We
projector?
don't,
at-
yeah, we don't need it. So yeah. Thank you. Hey, have a seat. No, please go ahead.
From far away. From far away.
Do we need the mic, or you think it's fine? We don't have enough.
Granted, slightly longer, but very coming up, yes.
Coming up. Hi, nice to meet you.
Nice to meet you.
Yeah,
Okay, so-
yeah. Share for you.
That's good.
Super Bowl conventions.
Okay, hi everyone. So welcome to this summary panel of today. And I would like to welcome again Kostas Helges. Thank you. Also our organizer, Albert, Danny, and also joining us, Grant from the University Lab of Ornithology. Thank you for coming. We know it's been tricky for you. So this is the third edition of the workshop, and we have seen today so many people. I think it makes it a timely moment to reflect on where are we going as a scientific community. So I would like to open a discussion and ask some questions for our panelists, but I also welcome your participation. So I would like to ask first question: what are the current challenges that CVPR Animals as a scientific community is dealing with? And what is the future vision for this? What do you think we should do as a community? And where do you see this community in 10 or 20 years from now? I know it's a bit difficult. Yeah. I think that Albert, in his final slide, made some comments which resonate with this question. So maybe we can start, Albert, with your vision.
Yeah, I think the big transformation is that the tools that we're developing are now easily turned into actual applications, which was not the case, let's say, 5-10 years ago. And now that in veterinary sciences, for example, people recognize the need to deal with more modern tools of observation. And practically, observation has been a bottleneck for them for many years. It takes a lot of effort. Annotation takes a lot of effort. This has been the same in psychology, obviously, but also in some of the veterinary science problems. So they look for opportunities to extend their toolboxes, which I think is a great idea. So that's what I think is at least for us in the field is going towards. And then we have a lot of experience working with humans, doing a lot of different tasks to understand human behavior at different scales. I mean, we look at individual scales, but we also look at interactions, so dyads or small group interactions. And then we also do computational social science, where we look at thousands and millions of people at the same time. And we have developed pattern recognition-based tools to address each of these scales. Now, I'm guessing that some of these tools are adaptable to problems of the animal kingdom as well.
Thank you. Any comments? Okay, Grant, would you like to go next?
Sure. So I haven't been here today. I've been in another workshop. So just a quick overview of who I am. I'm a researcher at the Lab of Ornithology at Cornell, and so I helped build the Merlin app, if any of you guys have used that, as well as I built the iNaturalist computer vision systems and Seek. So my experience is mainly working with citizen scientists. So yeah, I mean, you asked about kind of challenges, opportunities, where we're going. I think right now, like the biggest challenges I'm thinking about is how do you take these models that have been ultimately trained by citizen scientists and give them to scientists and build up trust in those scientists that the models can do what they originally were designed for. So what I see at the Lab of O is, we take sound ID, we want to apply it to thousands of hours or even more of audio. There's no way we can go through and ground truth the results. So how do you do this kind of active evaluation? in order to give the scientists some kind of reasonable expectations on what's working well and what's not working well, so they can draw kind of accurate conclusions. Because I do think like these models just kind of used in an inappropriate way can lead to incorrect conclusions being drawn from data. And so it's kind of our responsibility to make sure that we not only give them the model, but the tools to figure out how well these things are working on brand new data. So yeah, that's kind of like where a lot of my kind of current thinking is going, is tackling those challenges. And I think the future is just super exciting. I mean, I think all the tools are just going to be getting better. The speed at which we can analyze and collect the data is going to get better. And so we kind of it just reinforces this need of that ability to gain trust in all of this. And so, in 15, 20 years, I'm hoping that not only do we have kind of like well annotated data sets that can solve practical real world problems, but we've also kind of gained the support of not just the computer vision community, but ecologists and biologists from other communities are actually trusting and using our tools. So that's.
So you think citizen science is key in this vision?
I do. I think that they've proven their ability to help us annotate data. Of course, there's noise in there and there's bias in there, of course. But I think we've sort of chipped away at the problems of handling those biases. And so I think that is at least one way forward. Doesn't have to be the only way, but yeah.
Okay. Thank you. Kostas, did you want to comment?
Yeah, I would also mention the word trust. I think talking first, it is a great experience for computer science students and engineers to talk to real scientists, like biologists, ornithologists, because real scientists go through a lot of pain in order to produce data, which computer scientists have not experienced. And they have not experienced also that get the result might take 3 years, get just one journal paper. So in this sense, I think they regard it as naive. So I think I'm senior enough to say that our results were not good. The reason I say it's not good because really they're not usable in an automated way by the biologists. The way they are usable still is that because somebody already annotated what is true, they can use only the good stuff. And still, in terms of geometry, and, this kind of annotation, they are very helpful. But and they do observations for them. For example, a physicist, they build an experience with an instrument. This is a very painful process, right? An extremely painful process, which computer scientists are not used to. This is what this is a topic also in the third. I will talk again in the third sentence. But 80%, probably 90%, one of the best percentages I've ever seen today, still actually something that still has for a biologist or a physicist.
Thank you. Honey, can we get your perspective?
I have a lot to add to this. As a not a computer vision person who likes to use the algorithms that the community creates. In my mind, I mean, yes, it means a lot that these tools become easier and easier to deploy and use. But as we were building Ocean Vision AI, we did like very, very intense surveys and discussions like around user-centered design. So like, what were the real pain points that other marine scientists or people who are generating this visual data, like what were they dealing with? And if you talk to this community, it's not just the marine scientists, it's also like contractors that have to collect this visual data to evaluate whether some offshore activity could or could not happen. So like environmental impact reports. These are groups that just don't have a lot of available resources. So you just don't have enough people in your group with the expertise or the time to even spin up these much easier and simpler tools. That yes, it's simple for this community. It's not very simple for other communities. And so I think at least in my mind, this is something that I agonize over a lot, like with AdamNet and some other activities. It's like, how do you actually bring the experts together, right? How do you aggregate expert knowledge and have crosstalk between, you know, these groups or these organizations? Like, I would highly recommend, if you all are looking for use cases, to go either to the Ocean Sciences meeting, which is every other year, or the Society of Integrative and Comparative Biology meeting, which is every year. And these are groups that do phenomenal work in terms of data set generation, collection, instrument development. They know what the relevant use cases are, right? And just having that crosstalk between these two communities could, you know, really open up lots of opportunities. So yes, I agree. Like the citizen science stuff, the stuff you've been doing is just so inspirational, which is why we're trying to make a video game. But like, I think it's the connecting these expert user groups will be phenomenal if we can solve that problem.
Thank you. Any comments from the audience?
So I've been as a computer scientist working with biologists for the last 5 years. And one of the things that I observe most is that this crosstalk has one issue that most of the biologists, let's say, are still, I mean, when we say using machine learning, there is a very small group. A lot of them have actually problems which are 2D tracking, and it's sufficient to study what they want to study. But they say during the crosstalk, major problem they have is when you talk to a computer scientist, he or she, they're looking to publish in CVPR and ICCV, and they don't find those problems interesting enough because they are like, well, I can't get it published in a venue that I want to publish. So, sorry. And that's the general case. Or if the methods are developed, then they are not to a point where a biologist would be super easily using it. There's only one, I think one of the tools to be mentioned, something like deep lab cut or maybe mega detector, which is reaching that level. But most of the tools that are even working on these problems are left at a point where biologists have to figure out so much that they end up doing manual work. So where do you think this connection builds? Like how to do the problems, which then somebody doesn't have to worry about publication, and how does it work?
Thumbs up.
Okay, so this is actually a topic I think that came up a little bit during the Visipedia workshop at CalTech a couple months ago. And I mean, if you think about like kind of you bring back to mana, but then there's all these other challenges. Like, what do you do with your data? How do you host it? How do you organize it? Like, and so if you can help solve that problem, right? What that does is fix at least a data accessibility problem, right? So that's, I think, a big barrier between the two groups. Why we focus on FathomNet so much, because it's like, hey, if we can't get labeled data to you all, then you won't be aware of all the variety of use cases that computer vision could have in the ocean domain. So I think that's part of the reason why we focused on data infrastructure. And I think this is what I love to see, like with Visipedia or other groups, is like think about creation of data infrastructure that allow it to be a platform that's ripe for trying a whole bunch of experiments, right? It's not, so it doesn't become, you know, like an impenetrable like chasm that you have to cross on both sides. You're kind of creating something in the middle where, you know, there might be a science use case and they have a particular outcome that they want to see. And then within that same platform, you're then able to plug in all sorts of different algorithms or approaches like assisted learning or whatever to then solve it. So what happens maybe is that you get your data output, the actual need for the scientist or the researcher. And then for the computer scientist, you know, maybe some of the experiments that you need to run to then publish in the CVPR. I think there's so many different ways that you can approach that, but I feel like something like that would be very, very helpful. And already there was a report that came out maybe last week, the Committee for AI, where they were suggesting doing something like this, building kind of this infrastructure that allows people to get access to these tools, but also an aggregator for ideas where, you know, the community can try all sorts of experiments. I'm not saying that's the solution, but I feel like that is one way to bridge the gap. But, you know, it is tough, right? Because I think some of us work on very, very different timescales. If I can get a Science or Nature paper every 2 or 3 years, that's fantastic. I mean, that is really fantastic. I mean, yeah, there is that discrepancy between like how we're awarded. But I think if you can create structures to get us to join somewhere in the middle, I think that might be.
So a comment there?
I have a follow-up question, I guess, to that. Could you talk, so I guess I've thought about a lot of, when coming from similar places, so we thought about similar things, I guess. Could you talk more about, like, even what you were talking about sounded really nice about, like, building these platforms, I guess, to create these kind of integrations. My understanding is that to do that in kind of like a sustainable way, even that takes like at least a handful of software developers and people like that who just are going to be not just separate potentially to sort of academic researchers.
Yeah.
And can you talk about, I guess, if that's the case in your experience, and then also if it is, how that works in terms of just kind of funding and I mean, it feels like without being careful so often, you know, you can have these things that kind of last the funding cycle and then disappear. And so just how people think about having that additional work that is
Yeah, no, I mean, I think just following up, I think like there's like always decisions and trade-offs to be made, and you can be strategic in those partnerships that you strike with biologists and ecologists. I know Sarah and I have had great success with just finding folks who have those resources to hopefully get the databases running and managed for longer than just a CVPR cycle or an ICC cycle. So yeah, so our collaborations with the Lab of Ornithology and with iNaturalist and then Sarah with a bunch of camera trapping work has been like, you know, making like kind of being reasonable about the expectations on like what are our timelines for our conferences that we'd like to publish research at, and then what are their goals. And so it's some of this is just like kind of going back to like people communication type things of just setting expectations appropriately, finding those collaborations. You know, like there's a lot of people out there with problems to work on, but some of them are in a better spot to actually sustainably work on those problems, and you know, make sure that data is going to be around and that there's folks that can do the annotations for you. Yeah, and so you know, unfortunately, we can't work on everybody's problems all the time, so we do have to be strategic about which ones we approach. And then, you know, take advantage of those opportunities to then share the results with folks who don't have those resources, right? Getting those pre-trained models out there, getting those data sets out there to give them a leg up when they have the resources to take those steps. But yeah, I think it's just being kind of realistic with your time and resources and being strategic about those collaborations you set up. So.
Yeah, I wish Tanya was here because I feel like she would be able to speak a lot to sustainability. I mean, I can talk to our goals with Ocean Vision AI. But you know, we're essentially halfway through or more than halfway through a 3-year long design, development, and launch process. So it's very, very crazy. Have you heard of the NSF Convergence Accelerator? It's a new program that's out of the TIP directorate at the National Science Foundation. TIP is Technology, Innovation, and Partnerships. It's very, very different from other NSF programs where, you know, instead of just running a research program or, you know, trying to answer a particular research question, the outcome of this funding call is an actual product. It could be, you know, a software tool, it could be a website, it could be all sorts of things, but it has to address a relatively big societal challenge in the United States. And so that was actually something we proposed working on, was trying to solve this data infrastructure problem that a lot of the ocean science community faces. But the main driver for it is how we understand the growth of the blue economy. So there's a massive growth in wanting to, you know, do human-based activities in the ocean to exploit certain aspects of it. And the way that is currently being done now, it's being done without a lot of information and data. But not only do you have this need, you also have a potential source of funding. So what we basically pitched is this approach where through Ocean Vision AI, that's the FathomNet portal and a game, it's solving the data infrastructure challenge that a lot of people have. Could be based on subscription models, depending on who you are or how much data you have. But parts of it, like FathomNet and the game Fathomverse, are completely open and public. So through those other mechanisms, you can create engagement, re-engagement, perhaps hooks for philanthropy. Like iNaturalist, I believe, is funded like 50% with philanthropy, which I think is fascinating. And then balancing that philanthropy with, some of the, like a subscription, let's say, is what we're hoping to do. But I can't say it's going to work yet because we would officially launch the whole program next July, essentially. But so it's trying to figure out ways to, similar to your own like financial portfolios, come up with something that's, you've got options besides just one in particular.
I have a comment on the availability of data because so in my group, we work on computer analysis of human behavior at different scales. And the largest scales is that we look at migration behaviors and refugees. We work with mobile phone data. And then we realize that some of the technologies that are based on the data can be double-edged swords. So they can be used to help the refugees or also harm the refugees. One example is you have the application to find from the satellite the boats that are crossing the Mediterranean for helping these boats. But then it's also used for pushbacks by the countries. Now, imagine that you have a lot of ocean data available. And let's say I'm a big company looking for whales to hunt or do fishing in massive scale. And I look at some indicator like that, machine learning is very powerful. Maybe I can use some of the indicators, reduction in plankton concentration, to find large amounts of fish in an area, and then I go and fish them. So the risks are very difficult to assess, especially if you're a computer scientist working on a very specific computer vision or pattern recognition related problem. If you do not understand the social implications or implications of the technologies that you're developing. I mean, I'm imagining that the person who develops a recommender system algorithm for Twitter, can they have guessed what the echo chambers and filter bubbles will cause in the society and make all the radical approaches even more radical? Like that's so much in the future. But when these tools are becoming more and more available to people, it also changes the way certain things are done. So what are the ethical pitfalls there, like of this kind of technology? That I think is, I mean, for companion animals, we've been discussing this, right? I mean, you develop a simple robot to give your cat food when you're away. Will this, for example, make it easier on people who want to live the cat for a week, for example, with the robot at home? And will they actually end up damaging these pets' lives, right?
I would like to generalize and comment on that. Are ethics different for companion animals and sea life and bacteria that we saw? So if we discuss what is our common denominator here, when we work on such different species, I mean, even if we work on this animal ethics, what do we do about the diversity of the species?
Typically, you talk about harm, right? I mean, you try to balance benefits and harms, but I don't think anybody's trying to benefit harm balancing for bacteria. Right? I mean, for companion animals, we may do that, but also for maybe whales or interesting animals like tiger sharks or coral reefs. But are all sea animals or all animals as interesting? For example, can we build a citizen science project that will help cockroaches? I doubt that, right? I mean,
Okay, Helge, would you like to add your perspective?
Yeah, we're still thinking about the scale, which is somewhat related then to the scale of the impact you have as well. I wonder what are the things where we have to work together as a community. I see this breadth for research and smaller groups doing something for bees and fish and what kind of things. And the platforms we create now, it's definitely the right direction. Do we need something else as well? Do we need to think in our algorithms more to make it more scalable? That's I think one path forward. I don't know if the answer. It's one observation. What do we need to do is to work better as a group than individuals?
Just like related to same before revision conferences. So I can report that most of the time you get the comment or you consider only one species that it applies to other people. To answer, if you want to get your paper published, we need to work on generic and so, but I agree on having data benchmarks. So with clear benchmarks, I think and this is naturally chances to consider it and with the contributions.
They're saying we should run some challenges.
Yeah. Action items for the next year.
I would just love to say we should generate benchmarks, but we need to be very careful that we're generating them with the people who actually care about that species or that data and with a lot of nuance and context. Because I know I've seen a lot of benchmarks, computer vision benchmarks for biological data where it's just like you're memorizing it. Like some version of you're memorizing it and the model's not going to be useful. So I think we, particularly this community, we do want to think about how do you generate multi-species benchmarks? You know, how do you generate things where we're sort of trying to build, who knows, work towards something together? It should make sure that it's not like the aggregate at the detriment of any specific problem. And I don't really know if there's like a perfect way to do that, but just, we need to make sure the benchmarks are representative.
Yeah, there is a comment there.
I had a very specific question about synthetic data sets. We talked a lot about data optimization today, working with small data sets, or labeling very large data sets and how we can do that, as well as latent spaces, like these translational models, say like language vision or synthetic data in this real world. My question is synthetic data, is this ability to generate synthetic data, train translational models, is that something of shared interest in the community, or is that something that we expect a very like high effort, maybe lower support?
Anyone else? Any thoughts?
For the cases that I had, there's a very clear advantage, I think. How you would create synthetic data for the type of interesting deep sea creatures that we have seen.
Yeah, I think static data is great, but currently for what we have for humans, not and create static data set, static synthetic data set for each animal species. I think there is a paper here, CVPR, within James, where they are with so I think that's a very interesting direction where you generate the synthetic data you need, a simple tool that everybody can use. I think that's a very interesting direction.
I think it's very, very hard. We look for birds, and we talk to Pixar also. Most of the stuff is Pixar is really animated by hand. There is no really biomechanical process of the what size we can buy birds for $500. But these models are really very restrictive.
Okay, thank you. Thank you. Any comments? Yeah, go ahead.
One more thing,
Yes.
one more thing.
So I was thinking, if you release a bird into the Panoptic Studio, maybe you can get a full 3D. So there are some generic tools that kind of might give you the kind of data that you can actually then crop scale put into your. So maybe we need some new ways of generating synthetic data, not just generative to sale networks.
Actually, we did the opposite. Never did that. I did it all the way.
I have just one more comment on the data set challenge aspect. I think it's really an opportunity for computer science to not have just squared errors. Maybe the task is like explain the mating behavior of the birds. That's your task. And maybe in between you predict some key points, but they need to be good enough to when do the task. So, but maybe you come up with some other engineering which is better for the task. So having this end goal as the challenge maybe is the simple part.
I have one more comment. I mean, this is a real pain, the CVPR discussion. With every student, people want to work on animals. It's painful as you can get. But it's not painful for people like me who didn't grow up in this situation. But I have full understanding. I mean, I cannot start saying about this one anymore. So I cannot go into psychology about this. So I think the long term may be in biology, or if this is generally in AI for science, this would go AlphaFold or something like that. 90% of AI for science has lower number of applications. That's why we have so many workshops. Also, I think what would really ease the situation is that biology departments hire more computer science PhDs, not biology PhDs with computer science work. That would make this at least attractive for people who want to follow an academic career. And also make more attractive also publishing journals and really follow the long-term painful science research to do that. Again, publishing at CVPR, which is painful in a different way. So that's and really cultivate. I think this is a matter of culture. And it is up to us also who write on Twitter, etc., to cultivate the importance of working for science. For example, tell your students to work, to go to work to NASA, for example, right? For PhD students whose dream was to work to NASA. Even now, one of my postdocs went to CVPR. And really actually try to promote science results. Like the AR glasses from up on there.
Yeah, I wanted to add to that today. And I'm not a domain expert, but the whole time I was listening to the talk, I had this recurring question in my head, like, what real world problem does this actually solve? And I think if you do that before you start on any of these problems, I think that's so important. Because there are whole communities, groups of researchers, so resource constrained that could not imagine what you are able to do with the algorithms that you've created. And you can be solving very timely real world problems with them if you make that connection. So please just start with answering that question first before diving deeply into things.
I feel like something that's been brought up in my experience as well is that, so I guess going off of this is that there are all these AI developments that we're coming up with computer vision. But in science, I feel like oftentimes it's not even the algorithm that's the bottleneck, just the interface to it. So I guess I'm curious about your perspective on where this human-computer interaction research between like bridging these two gaps fits into academia as a whole, and like how should we conduct it?
So Brent, you probably have something to say to that.
Yeah. So I think it's incredibly important. Yeah, I think that's kind of this like next phase of challenges that we should start thinking about. So get models that are working on our benchmarks, and then try to get them in the hands of experts that can use them. The publication venues for that type of research may or may not be CVPR, but it certainly could be other venues like Chi or even Dub, or one of the knowledge communities. Like, yeah, I mean, I think we just need to be a little bit more open of like where we're going to be trying to send this work. Like, yeah, CVPR and these other NeurIPS and these other places are always going to be this kind of like challenge to get some of this more applied work into. And so I think we just need to be like more open to exploring some of these different conferences. But yeah, absolutely. I mean, I think that the challenge of how do you, and again, this kind of goes back to this trust, like, I think we've built models that are useful, and now how do I convince someone like Akani that they are really useful? I mean, that's like both using the model and presenting the results in a way that she can understand, in a way that she can interact with. And then, I mean, the thing that gets me excited is like, often when you show someone like Akani these type of results, she immediately wants to give you feedback, right? So like, how do you then take that feedback and actually, yeah, they're very easy to criticize, or quick to criticize, I should say. So like, yeah, how do you get that stuff back into the system and improve it? I think it's that next step of this progress, which is, it's exciting that we've gotten to that point, right? I mean, certainly 10 years ago, this was not the case. But yeah, now it's like we get to start thinking about these next set of challenges.
Yeah, I think actually this workshop is incredibly important for creating a venue where you can explore this kind of. So I think we should aim at making this conference, maybe like the face and gesture was a workshop.
Nice idea.
Yeah, on that note, we're always open for more co-organizers to give you ideas. To run a challenge or whatever you think, and let us know, reach out. If you want to nominate somebody else too, who you think would be very suitable, send up the name, we will find them. So yeah, happy to grow.
Maybe we can start with an email list, kind of build community. That's, I think, always a good place to start.
Yeah, certainly we can do it.
Yeah, I will create an email list and put it on the CVPR notes webpage, so you can all register and connect further.
There's also the AI for conservation slants. Great.
Yeah,
We can use that too, maybe.
Yeah, go
maybe
for it.
there's more.
It's already 1,000 researchers plus globally working on these types of problems for animals, usually in the context of conservation, but it's a growing community. So feel free to go on there and make yourself a subgen.
Okay, sounds good.
Yeah. Okay, on that optimistic note, we thank our panelists. Thank my wonderful co-organizers, and now we are headed to the posters. Thank you, and see you next year.
Yeah. Oh no, thank you so much for inviting me. Yeah. I'm not sure if I contributed anything, but yeah. No, that's very cool. I know it's always like I wish one of these was on like a different day. Like, yeah. Oh, thank you. No. It's awesome to be here.